Good afternoon, learners. Welcome you all to the Gyan Darshan session. Today we will take up one topic, that is extracellular matrix. If you remember, in one of my earlier sessions, I covered on cell junctions. I mentioned in that session that I will be taking up the next part, that is extracellular matrix, in my next discussion. So today we will be taking up this topic, extracellular matrix, and it's a part of your MSc Zoology course. That is, you have a course on molecular cell biology, and there you have one unit on cell junction and extracellular matrix. So we have already discussed about cell junction in our earlier session. Today we will be taking up the next part, that is extracellular matrix. Now to start with, what is meant by extracellular matrix? Extracellular, that means which is present outside the cell. So you see that this particular component, that is extracellular matrix, also has a very important part to play in maintaining a proper structure of a cell in order to maintain the normal function of a cell, not only structure but also the function of a cell, stability of a cell. So all these are equally regulated by this extracellular matrix. So when we discuss this component, then we will have to take a look at what are the different constituents of this extracellular matrix, or what is this ECM. In abbreviation we call it ECM. What is this ECM composed of? So if we go to our first slide, that it is composed of a variety of proteins and polysaccharides secreted locally and assembled into an organized meshwork. So basically you have two components, two main components which we will discuss. One is it has the polysaccharides or the carbohydrates, and the second the proteins. So the first part of my discussion, we will be focusing on the polysaccharides or the carbohydrate part, which are mostly called as the proteoglycans, and among the proteoglycans you will find the glycosaminoglycans as the major component of extracellular matrix. Okay, and these are in abbreviated form. Glycosaminoglycan, we refer to them as GAGs, GAG, that is glycosaminoglycan. And the second component, that is protein. So with respect to the proteins, you have the different proteins like collagen, elastin, fibronectin, laminin. So these are the four main proteins which are components of ECM. So today we will be discussing one by one. First we will take up glycosaminoglycan, their characteristics. Then we will go to the structural components of the collagen, elastin, fibronectin and laminin. And finally we will see that how the extracellular matrix actually regulates the stability of a cell. What will happen if the extracellular matrix gets destroyed? Then what will be the fate of the cell? So you should not think that since it is present outside the cell, it has no major role to play with the cell. In fact, it has a very, very important role to play with regard to the proper stability, with regard to the proper structure and the function of a cell. So the vertebrate extracellular matrix was once thought to serve as a scaffold. That means initially it was thought that it is just it just plays a role in maintaining the structure. But now it has been shown that it has a more active and complex role in regulating the behavior of the cells, in regulating the survival, development, migration, proliferation, shape and function of a cell. So it has a tremendous role to play with regard to the functioning of a cell, not only the structure but also the function. So two main macromolecules, as I said, they are the components. One is glycosaminoglycan or GAGs, which are covalently linked to protein in the form of proteoglycans. So what happens in a ECM is you have the proteoglycans, that is the GAGs, they are linked to the protein. And what are those proteins? You have the different fibrous proteins including collagen, elastin, fibronectin and laminin, as I have already mentioned. Now we take up the first component, that is the glycosaminoglycans or the proteoglycans. So if you see the diagram, you can see that the GAGs basically they have some repeating disaccharide sequence. Like here they have taken the example from dermatan sulfate, and you can see that they have a repeating disaccharide unit, which in this case is iduronic acid and N-acetyl galactosamine 4 sulfate. So what is proteoglycan basically? Proteoglycan is, it forms a highly hydrated gel-like ground substance in which the fibrous proteins are embedded. So within the proteoglycan, the fibrous proteins like collagen, elastin, these are all embedded. And what is the function of the polysaccharide gel? It resists compressive forces on the matrix while permitting the rapid diffusion of nutrients, metabolites and hormones between the blood and the tissue cells. So on one hand, it is maintaining the structure, it is resisting the compressive forces, and on the other hand, it also has role in the diffusion of nutrients, metabolites and so on. So it has two functions. So the basic structure, as you can see in the diagram, that they are typically 70 to 200 sugar long, and there is a very high density of negative charge, because see the acid, iduronic acid, that means any acid, it has the COOH, that it has the carboxyl group. So COO is minus, and here also you find N-acetyl galactosamine 4 sulfate, that is again sulfated, which means that again there is a negative charge. So all these glycosaminoglycans, they are highly negatively charged. This is one of the very important feature of the glycosaminoglycans. Now what is the physiological significance of this? We will very shortly we will understand that why these kind of components are so high. They are so high in their negative charges. What facility does a cell get out of this? Now, glycosaminoglycans are unbranched polysaccharide chains composed of repeating disaccharide units. They are called GAGs because one of the two sugars is always an amino sugar, glycosaminoglycan. So, there is one sugar that is always an amino sugar like N-acetyl glucosamine or N-acetyl galactosamine. But again, as you have seen in the previous diagram, that this amino sugar is sulfated. Okay, N-acetyl glucosamine sulfate or N-acetyl galactosamine sulfate. And the second sugar, it can be any uronic acid. It can be glucuronic acid or it can be iduronic acid. So the ultimate feature is that because of the presence of the sulfated groups or carboxyl groups, these kind of components are highly negatively charged. Okay. Now what is the immediate significance of this? Immediate significance of this is that because they are highly negatively charged, they can attract cations. Like they can attract a cloud of cations, most notably sodium, that are osmotically active, causing large amounts of water to be sucked into the matrix. So they can draw a huge amount of water because they are osmotically active. This creates a swelling pressure or turgor that enables the matrix to withstand compressive forces, and therefore they help in the resistance of the compressive forces. So if somebody asks you the question that why the glycosaminoglycans can resist the compressive forces, the answer will be that because they have a very high amount of negative charge because of the presence of the iduronic acid, uronic acid and the amino sugars. That is why they are negatively charged, and therefore they can attract a huge cloud of cations including sodium that enables an osmotically active property to these components, and therefore they can draw a huge amount of water, and therefore this creates some sort of mobility and it helps in the withstanding of the compressive forces. So this is a very, very important property, and or you can say function, and the function is directly correlated with the structure. Okay, so this has to be understood very properly. Now what happens in case of a plant cell? Now in case of animal cell this is very clear, but do the plants or do the invertebrates, they also have this extracellular matrix? The answer is of course yes. And in case of plants, you see that you have the cell wall, and again this cell wall actually constitutes the extracellular matrix because the component of a cell actually starts from the plasma membrane. But in case of plants, outside the plasma membrane you have the cell wall, which is mostly composed of cellulose, hemicellulose, pectin, and also you have some structural proteins. So you have the ECM or the extracellular matrix also in case of plants. And in invertebrates, like for instance in case of crustaceans and insects, you have the chitin. So they have the exoskeleton, chitinous exoskeleton. Like in arthropods, they have the chitinous exoskeleton. So that is actually a part of the extracellular matrix. So this is present not only in animals but also in plants or in some lower groups of animals, like as I said, in the crustaceans, in the insects or the arthropods. Now see, if you take a look at this diagram, then you can see that how the GAG, that is the glycosaminoglycans, are actually connected with the core protein. Okay, so you see there are some link tetrasaccharide. The link tetrasaccharide actually consists of xylose, galactose, again another galactose, and the glucuronic acid. So if you see the chemistry, then you can see that the GAG, n number of GAGs, that is, we don't know how many GAGs, there are so many GAG chains, it's a huge GAG chain, and n number of GAGs, they are actually connected to the core protein, okay, through a link tetrasaccharide. And what is the amino acid, okay, that is involved in this linkage? That is serine. So the GAGs are connected to the serine residue of a core protein through a link tetrasaccharide, and that link tetrasaccharide consists of xylose, galactose and glucuronic acid. So you see that this linkage with the core protein is also very specific. I mean, to which amino acid it will link and how will it link, okay, through this link tetrasaccharide unit, that is also very conserved, because some conserved sugars are actually a part of this link tetrasaccharide. So you can see that in this way the entire GAG chains along with the core protein is associated, and this together constitutes the proteoglycan. So proteoglycan is thus a major component of the extracellular matrix, which is composed of the GAGs, as I said, linked with proteins. Now here again we will next come to another typical, you can say a simple GAG, that is hyaluronan. Now in case of hyaluronan, you can see that the repeating disaccharide is bit different. Here you see that it is glucuronic acid and N-acetyl glucosamine. But what is the difference? If you just see the structure, you can understand the difference, that in this case the N-acetyl glucosamine is not sulfated. In the earlier case, the amino sugar was sulfated in case of typical GAGs. But in case of hyaluronan, you see the first sugar is uronic acid. Second sugar is also the amino sugar, but it is not sulfated. So this is one major difference with the typical GAGs. So hyaluronan is the simplest of GAGs. It consists of a regular repeating sequence of up to 25,000 non-sulfated disaccharide unit. This is an important difference with the typical GAG. That is why many people, they do not consider hyaluronan to be a typical GAG because of this difference in structure and chemical composition. Okay. And in contrast with all of the others, it contains no sulfated sugar. All its disaccharide units are identical. Its chain length is enormous and it is not linked covalently to any core protein. So if somebody asks you what is the difference between hyaluronan and a typical glycosaminoglycan, your answer will contain 4 points. I repeat those 4 points. No sulfated sugar. Number 2, all its disaccharide units are identical. Number 3, its chain length is enormous, thousands of sugar monomers. And number 4, it is not generally linked covalently to any core protein. Another difference, in fact there are 5 points. Another point is that the GAGs are synthesized inside the cell and released by exocytosis, whereas in case of hyaluronan, it is spun out directly from the cell surface by an enzyme complex embedded in the plasma membrane. So 5 differences are there which you should remember, and if you are asked, then you should be able to write those 5 points. Now, proteoglycans are usually distinguished from other glycoproteins. Now, we come to another point, that is, can you regard proteoglycan to be a glycoprotein? What is the difference between a proteoglycan and a glycoprotein? Apparently, it might seem that these are same, because glycoprotein also is composed of glucose and protein. And proteoglycan, glucose or any sugar, I am talking of sugar and protein. Whereas proteoglycan, same, that is protein plus that is the sugar or any carbohydrate. But there is a basic difference. What is that difference? Difference is glycoprotein, it contains 1 to 60% carbohydrate by weight in the form of short branched oligosaccharide chains. But proteoglycan, they contain as much as 95% carbohydrate by weight. So in case of proteoglycan, actually the carbohydrate component is more. Protein component is much lesser. So 95% they are carbohydrate by nature. Only 5% they may be composed of proteins. Whereas in case of glycoprotein, they are 1 to 60% carbohydrate and the rest they are proteins. So glycoproteins, they have a much larger protein composition as compared to the proteoglycans. Proteoglycans can be huge. Now there are different proteoglycans in different tissues, like cartilage you have aggrecan. Okay, then so based on different tissues you have the different types of proteoglycan chains or the GAG chains. Now what are the function? The GAG chains can form gels of varying pore size and charge density. Hence, they can serve as selective sieves to regulate traffic of molecules and cells. Heparin sulfate proteoglycan called perlecan has this role in basal lamina of kidney glomerulus, which filters molecules passing into urine from the bloodstream. Then they can also take part in chemical signaling between the cells. They bind the various signaling molecules. And the third point, the ubiquitous growth regulatory protein transforming growth factor beta, which in short we call TGF beta, binds to the core protein of several matrix proteoglycans including decorin. So from these points you can well understand that the proteoglycans or the glycosaminoglycans, they have a major role to play also in the cell signaling or the chemical signaling process of a cell. So in that way they are very vital component. Now here if you take a look that some common proteoglycans have been listed, like as I said, in different tissues you have different proteoglycans. Agrican, that is chondroitin sulfate, that is chondroitin means the cartilage. Beta glycan, this is present in cell surface and the matrix. Then in decorin, that is it's distributed in connective tissues. Perlecan, this is in basal lamina. We will talk about basal lamina also in the later half. Syndecan, this is epithelial cell surface. Dali, that is in Drosophila, that is again present in the cell surface. And see, each of them, they have their own functions. These functions are mostly connected to the cell signaling process also. Now we come to the next part. So first part we have discussed, that is first part or the first component we have discussed, that is about the proteoglycans, that is about the glycosaminoglycans, their structure, their chemical composition. Now we come to the second part or the second component, that is the proteins. Now when we talk about protein, first of all we will start with the collagen. Okay. So some types of collagen and their properties. So you see the collagen molecule, that they are actually divided or they are actually categorized into 4 groups. One is fibril forming collagen or the fibrillar collagen. One is fibril associated collagen, network forming collagen, and transmembrane or other collagens. Most abundant or the most you can say the common collagen, that is fibrillar collagen. So you can see that the collagen is divided into different types, type 1, type 2, type 3, type 5, type 11. So these are all the fibrillar collagen. Then fibril associated collagen 9, and then you find 12. These are fibril associated collagen. Network forming collagen, that is type 4 and type 7. Transmembrane collagen, that is type 17, and others, that is type 18. If you see their distribution, then fibrillar collagen, you see that in bones, skin, tendon, ligaments, cornea. Okay. So different, they are widespread in distribution. If you see the fibril associated collagen, they are also present in cartilage, tendons. Network forming collagen in basal lamina. Transmembrane collagen, that is hemidesmosomes. Okay. Among the others, they are in the basal lamina, then around the blood vessels. So these are the 4 main categories of collagen. So we won't be discussing too much details on that, but the fibrillar collagen, that is the most important, and we will discuss the structure of the fibrillar collagen, and then we may take a look at the difference between the fibrillar collagen and the fibril associated collagen. So these 2 points we can take up and we can have a discussion on this. Now first of all, if you see the structure of a typical collagen molecule. So this is actually the structure, as you can see in the diagram. Now what does this diagram show? Or if somebody asks you that what are the salient feature of a collagen molecule, I mean salient features of the structure of a collagen molecule, then what are the points that you are going to mention or write? Number 1 is this collagen molecule, it is about 1000 amino acid long, number 1, and number 2, it is a left-handed helical structure. Very important, left-handed helical structure. 3 amino acids per turn, and glycine is present at every third amino acid position. So, every third amino acid of this molecule is a glycine. And you all know that glycine is the simplest amino acid. So, what are the characteristic points? Number 1, 1000 amino acid long. Number 2, left-handed helix, left-handed helical structure. Number 3, 3 amino acids per turn. Number 4, glycine at every third amino acid position. So, these are the 4 major points that your answer should always contain. Now, an alpha chain is composed of a series of triplet, that is gly-XY. XY can be any amino acid, but gly is always fixed, as I said, because third amino acid is glycine. The other 2 is, it could be XY, means it can be any amino acid. X and Y can be any amino acid, although X is commonly proline and Y is commonly hydroxyproline. So, the collagen molecule will have glycine as a compulsory component, compulsory amino acid, and other than that, it has these X and Y amino acid, which can be any amino acid, but more commonly proline and hydroxyproline. So, now we will see that why they have this glycine. What is the advantage that glycine conferred to this kind of structure, that we will see. So, collagens are a family of fibrous protein found in all multicellular animals. They are secreted by connective tissue cells. Now, the collagen is actually present in an inactive form, or it is present in when it is first synthesized, it is actually present in the form of the tropocollagen, and later on this tropocollagen, it gets converted to a matured collagen molecule. This also has to be remembered. So, when it is a matured collagen, as I already said, that it's a long, stiff, triple-handed helical structure. 3 collagen polypeptide chains called alpha chains are wound around one another in a rope-like fashion. They are rich in proline and glycine. Also, hydroxyproline may be present. It is a triple-stranded helical structure. Now, why proline and why glycine? Now, proline, because of its ring structure, stabilizes the helical conformation in each alpha chain, and glycine, as I said, is the simplest amino acid and smallest amino acid. Therefore, glycine allows the 3 helical alpha chains to pack tightly together to form the final collagen superhelix. So, if somebody asks you that why proline, why glycine, why they are so abundant, why they are present in so huge amount in the collagen molecule, I hope you will be able to answer. Because of the ring structure of the proline, and because the glycine is the simplest and the smallest amino acid. That is why it allows the proper packing, easy packing. And in case of the proline, because as I said, it has the ring structure, so it stabilizes the helical conformation of the collagen molecule, because it is a triple stranded helical structure. So, if you have this background in mind about the structure of the collagen molecule, next, what are the functions? Now, collagen fibrils, they form structure that resist tensile forces. In contrast to GAG, when we discussed about GAG, we said that they resist the compressive forces. Collagen fibrils, they resist the tensile forces. So, this is the difference. So, you see that why GAG is also important and why the collagen fibrils are equally important. In tendons, they are organized in purple bundles aligned along the major axis of tension. In mature bone and in cornea, they are arranged in orderly plywood-like layers with the fibrils in each layer lying parallel to one another. So again, these collagen molecules are also present in different types of tissues like tendons, bone, cornea, etc. So I just present the summary, like you see the fibrillar collagen type 1, 2, 3, 5, 11, fibril associated collagen type 9, and then you have the type 12, network forming collagen you have type 4 and type 7, collagen like, that is type 17. Now, what is the difference between the fibrillar collagen and fibril associated collagen? So, how the fibril associated collagen differ from fibrillar collagen? So, these are some of the points. Their triple stranded helical structure is interrupted by one or two short non-helical domains, which makes the molecules more flexible than fibrillar collagen molecule. Number 2, they are not cleaved after secretion and therefore they retain their propeptides. That is, as I said, the tropocollagen. Tropocollagen, then the more matured or after their maturation they get converted to the collagen. They do not aggregate with one another to form fibrils in the extracellular space. Instead, they bind in a periodic manner to the surface of the fibrils formed by the fibrillar collagen. Like, for example, type 9 molecules, they bind to type 2 collagen containing fibrils in the cartilage, the cornea and the vitreous of the eye. Whereas, type 12 molecules, they bind to type 1 collagen containing fibrils in tendons and various other tissues. So, basically their function is more to associate. That is why they are called fibril associated collagen. So, they are more associated with the fibrillar collagens. Now, we come to the next protein. So, collagen is over. The first protein, that is the collagen. Now, the next protein that we are going to discuss is the elastin. Now, from the very name it is clear that elastin means elasticity. So, the main function of elastin is to provide the elasticity. Many vertebrate tissues such as skin, blood vessels, lungs, they need to be strong, but they need to be elastic in nature. Particularly lungs, blood vessels, they need to show the elasticity. Who will convert this elasticity? These elastin proteins. The main component of elastic fiber is elastin, a highly hydrophobic protein which, like collagen, is unusually rich in proline and glycine, but unlike collagen, it is not glycosylated and contains some hydroxyproline but no hydroxylysine. Now, in this context, I would mention that when we have studied the collagen molecule, we have discussed that they have proline, they have hydroxyproline and they have glycine. Another thing which you should note is they may also have hydroxylysine. Okay? But when you talk about elastin, then in case of elastin, okay, if somebody asks you what is the difference between elastin and collagen, what is the similarity? Similarity is that it is like collagen, they also have proline and glycine. What is the difference? Difference is that unlike collagen, they are not glycosylated and they never contain hydroxylysine. They may contain hydroxyproline, but they never contain hydroxylysine. So, these are the similarities and the differences between elastin and collagen. Now, again, in case of collagen, in case of elastin, they are present in a earlier form of elastin is the tropoelastin, that is the precursor of elastin, you can say. They are secreted into the extracellular space and assembled into elastic fibers close to the plasma membrane, generally in cell surface enfoldings. After secretion, the tropoelastin molecules become highly cross-linked, generating an extensive network of elastin fibers and sheets. So here we are actually discussing that how the elastin molecules are synthesized. So initially it is the tropoelastin. Tropoelastin is secreted in the extracellular space, okay, and then after secretion they become cross-linked, the extensive cross-linking, and they form an extensive network of elastin fibers. Now, how these cross-links are formed? The cross-links are formed between lysine by a mechanism that it's actually the same mechanism that actually operates in case of the collagen molecules. So, the extensive cross-linking occurs between the lysine, which is also another amino acid, okay. So, that extensive cross-linking, it finally gives rise to a matured elastin molecule. If you see this figure again, you can see that how the elastin is actually helping in this controlling this stretching and relaxation mechanism. So, it is almost like a rubber band, the same mechanism like you can see like you find in case of a rubber band, that is stretching a network of elastin molecules. The molecules are joined together by covalent bonds which are marked with red or pink, whatever you say, to generate a cross-linked network. In this model, each elastin molecule in the network can expand and contract as a random coil. So, sometimes there is a stretching and sometimes there is a contraction. Just like in case of a rubber band you have, you can stretch it out and again you can relax it. So, it is the same kind of mechanism. The elastin protein is composed largely of two types of short segments that alternate along the polypeptide chain. The hydrophobic segments, which are responsible for the elastic properties of the molecule, and alanine and lysine rich alpha helical segments which form crosslinks between adjacent molecules. So, what are these crosslinks made up of? Crosslinks are mostly made up of amino acids like alanine and lysine. Now, elastin polypeptide chain, like the polymer chains in ordinary rubber, adopts a loose random coil conformation, and it is the random coil structure of the component molecules cross-linked into the elastic fiber network that allows the network to stretch and recoil like a rubber band. So, I hope you got the point that how this stretching and relaxation mechanism operate in case of an elastin molecule. And this is very important for proper functioning of the blood vessels and the lungs. Even in case of artery, like say supposing the largest artery that we have, the aorta, we call it aorta in case of animal systems. Now, if there is a mutation in an elastin gene, because elastin is a protein, so you all know that if it is a protein, there must be a gene, there must be an elastin gene. Now, if there is a mutation in this elastin gene, then it is found, like they have experimented in mice, or even if you do it in human, then if you see that if there is a mutation in the elastin gene, then it causes a deficiency of the protein in mice or humans, resulting in the narrowing of aorta or other arteries as a result of excessive proliferation of smooth muscle cells in the arterial wall. So, what will happen in this case? There will be rupturing. The normal elasticity that will be lost, and there will be rupturing of the aorta. So, there will be extensive damage to the arterial wall because this stretch and relaxation mechanism is lost. The elasticity is lost, and so there will be major damage to the arteries. Elastic fibers are not composed solely of elastin. Apart from that, they also have some other component, that is, the elastin core is covered with a sheath of microfibrils, each of which has a diameter of about 10 nanometer. The microfibrils are composed of a number of glycoproteins, like one such glycoprotein, which is a large glycoprotein called fibrillin. And this fibrillin, it binds to elastin. See that fibrillin is a different protein. Okay, elastin is a extracellular matrix protein, whereas fibrillin is something different. It is not a component of ECM, it's a glycoprotein. But if there is a mutation in the fibrillin gene, then it leads to a syndrome called Marfan's syndrome, which is a human genetic disease affecting connective tissue that are rich in elastic fiber. And that will also cause a major damage to the aorta. It will lead to the rupturing of the aorta. So what I mean to say is that, see, not only the ECM proteins are important, but the proteins which are associated with these ECM proteins, they are also equally important. Fibrillin is not a ECM protein, but it associates with an ECM protein, that is elastin. And if there is a mutation in the fibrillin gene, the effect is as severe as having a mutation in the elastin gene, because ultimately it will have the same consequence of causing rupturing of the arterial wall. So how delicate the mechanism is, that I think you are able to understand. Now we come to the another component of the, or rather the third protein, that is fibronectin. So we have two more proteins left, that is fibronectin and laminin. So if you see the structure of fibronectin, it is again a dimer. We call it a fibronectin dimer, and at the carboxyl terminal portion, the each monomer is joined by a disulfide bridge. So how this dimer is formed? The dimer is formed by the association of the monomers at the carboxyl terminal end through disulfide bridge. And the fibronectin structure, it has several domains. It is a multi-domain protein, and each of these domains actually have different function. Like there is a self-association domain, there is a collagen binding domain, there is a cell binding domain, there is a heparin binding domain. So it's a multi-domain protein, each domain having a major function, unique function, and they are the monomers are joined at the carboxyl terminal end through disulfide bridge. So these are actually the characteristic feature. And another important point is that these structures, they have a conserved amino acid sequence which we call RGD. RGD means arginine, glycine and aspartic acid. So these are the three conserved amino acids which are present as a in case of this fibronectin structure. So the extracellular matrix contains a number of non-collagen protein that typically have multiple domains, each with specific binding sites. And as we said, that among them is the fibronectin. It's a dimer with two very large subunits joined by disulfide bonds. And what you can do is that if you subject this fibronectin to a proteolytic enzyme, then you can see that they have the different domains. And each of these domains, as I said, they have different function. Like one domain can bind to collagen, another domain can bind to heparin, another domain can bind to specific receptors on the surface of various types of cells and so on. And they have a specific tripeptide sequence which is arginine, glycine, aspartic acid or RGD, which is found in the type 3 repeats. So I think these are the points which you should actually remember when you describe the structure of a fibronectin. Now, the fibronectin fibrils on the cell surface are highly stretched and under tension. The tension is exerted by the cell and is essential for fibril formation. Now, they have taken one example like uteroglobin. Uteroglobin, what happens is it binds to fibronectin and prevents it from forming fibrils in the kidney. So, if you have mice that have mutation in the uteroglobin gene, it accumulate insoluble fibronectin fibrils in their kidneys. So, that will cause a major damage to the kidney. So, the function of a fibronectin is actually proper functioning of the fibronectin or the maintenance of proper structure of the fibronectin will depend upon certain proteins like certain other proteins that bind to fibronectin. Like for example, in this case it is the uteroglobin. So, even in case of a little while ago we saw in case of elastin also. So, not only these proteins are important, but the proteins which are associating with these proteins, they are also equally important to maintain their structure and to maintain their function. Okay. So, now we come to the fourth kind of protein, that is laminin. So, fibronectin is over. Now, laminin. Where is laminin present? Laminin is actually present in the basal lamina. What is basal lamina? Basal lamina is beneath the epithelial tissue. The extracellular matrix can be organized as a thin tough sheet which we call as the basal lamina. And here we mostly find the type 4 collagen molecules. They assemble into a sheet like meshwork to help form the basal lamina. So, in basal lamina you have the type 4 collagen, but apart from that you have some other proteins also, and that is laminin, which we are going to discuss now. So, they have a more flexible structure than the fibrillar collagen. These molecules associate by their carboxyl termini to form head-to-head dimers. So, this is actually the structure you can say of a basal lamina. See, there are so many, you can see from the diagram, there are so many components. There are so many interactions. So many proteins are involved. Like as I said, type 4 collagen, laminin, then there is another protein called nidogen, then there is the proteoglycan also, that is perlecan. So, it's you can say it's a huge kind of assemblage of different components, that is the proteoglycan, the proteins like collagen, laminin, etc. Of course, you will not find elastin here, you will not find fibronectin here. You will find some collagen, type 4 collagen, and the laminin that we will discuss now. So, this is the structure of a laminin. So, it is one of the first ECM proteins which is synthesized in a developing embryo. Now, what is the structure of this laminin? The subunits of a laminin 1 molecule. It's a multi-domain glycoprotein composed of 3 polypeptides, alpha, beta and gamma, that are disulfide bonded, and they form an asymmetric cross-like structure. It is a cruciform structure. Okay, and each of the polypeptide chains is more than, if you take a careful look at the picture, you see it's a cross-like structure, crucified kind of structure. We call it cruciform structure. So, each of the polypeptide chains, so the proteins, the different chains are folded in such a fashion that they are forming a cross-like structure. So, each of the polypeptide chain is more than 1500 amino acids long, 5 types of alpha chains, 3 types of beta chain and 3 types of gamma chains. So, they can actually assemble and they can give rise to almost 45 isoforms or 45 combinations can be possible. So, that is 5 into 3 into 3. So, 45 kind of combinations are possible, and so that there are 45 possible isoforms available for laminin. Several such isoforms have been found, each with a characteristic tissue distribution. So this is actually the structure, and then the basal lamina, they have diverse functions. Like in the kidney glomerulus, an unusually thick basal lamina acts as a molecular filter, preventing the passage of macromolecules from the blood into urine as urine is formed. So you all know that in case of kidney, you have the glomerular filtration. This is one major mechanism. So that filtration mechanism is well controlled by this basal lamina. And because laminin is a component of this basal lamina, so you can well understand that the laminin also plays a major role in this connection. Then there is one kidney disorder, that is Alport syndrome. What is this Alport syndrome? It is a human hereditary kidney disorder. What happens here? Now in this case, type 4 collagen also has a role, as a human hereditary kidney disorder results from mutation in type 4 collagen alpha chain genes. So laminin is important. At the same time, collagen, that is type 4 collagen, as we have seen in case of basal lamina, they are also equally important, because if you have a mutation, then it will lead to this kind of disease, or rather syndrome in the kidney. As I have said that what is the function of basal lamina now, one can well understand that it is a selective barrier to the movement of cell. This basal lamina is present beneath the epithelial tissue. So they mainly function to prevent the fibroblasts in the underlying connective tissue from making contact with the epithelial cells. So basically the basal lamina acts as a partition between the epithelial tissue and the connective tissue. Otherwise, what would have happened if basal lamina was not there? The epithelial tissue and connective tissue, they could have got mixed. The different proteins, okay, which are present in the connective tissue, they would have got admixed with the epithelial tissue. So it acts as a very sheet of partition between the epithelial cells and the connective tissue cells. It does not, however, stop the macrophage, lymphocytes or nerve processes from passing through it. Because why? Because say supposing if there is a damage in the epithelial tissue, then you need the components to repair the damage. If there is a puncture, or if there is somewhere in the wound in the skin, surface of the skin, it needs to be healed up. So who will provide? Who will provide the component? All the components will come and then they will do the healing. So who will do that? Then the connective tissue is there. So that passage should be there. That is, from the connective tissue to the epithelial tissue, it should come. So it does not stop that. It does not stop the macrophages or the cells which are responsible for defense. It allows those molecules, okay, to pass through the basal lamina, and finally they come to the surface and then the healing is done. So that is also an important function of basal lamina, that it allows this kind of macrophage or defense cells to come to the surface. When tissues such as muscles, nerves and epithelia are damaged, the basal lamina survives and provides a scaffold along which the regenerating cells can migrate. So I hope you got the function of you could understand the function of the basal lamina. And I just repeat that the basal lamina function is well regulated, or the structure is regulated by the laminin and the type 4 collagen molecules. Now another very important component, or what should I say, that if the extracellular matrix gets damaged, what will happen? Because all these proteins, collagen and all, supposing these proteins are degraded, or these proteins are damaged, or these proteins are lost, then what will happen? The cell will lose its attachment to the substratum. That is, the cell will lose attachment to the extracellular matrix. Then what will happen? It will lead to metastasis. It will lead to, because they fail to adhere to the substratum. Okay, we call it anchorage dependence. If this anchorage dependence is lost due to the degradation of the extracellular matrix, then the cells will metastasize, the cell will undergo, they will not be attached, and this will lead to cancer, because we know that in case of cancer there is a metastasis, there is the spread of cells. Cancer cells never remains at one position, they migrate, that is why it spreads so rapidly. So one of the reasons for that is due to the degradation of the extracellular matrix. And why this degradation occurs? This degradation may occur sometimes due to the release of certain enzymes which are called the metalloproteases, MMPs, matrix metalloproteases. So these enzymes, what do? They actually degrade the extracellular matrix proteins like collagen, and so the stability is lost, the extracellular matrix gets degraded, therefore the cells, they lose attach to the substratum, the cells lose attachment to the extracellular matrix, so the cells tend to move, they don't tend to remain stuck at one position or stuck to the substratum, they tend to move, they tend to spread. Okay, so the metastasis occurs, and therefore they might lead to the cancerous effect. So you can see that how the extracellular matrix is important, why its stability is so important in maintaining the proper structure and stability of a cell. Finally, I will just discuss one more component in another 2 or 3 minutes before we close this session, that is integrins. This is another kind of protein. This integrin is actually an extracellular matrix. I would say it is a transmembrane protein. What is the function of this integrin? Integrin is the protein that actually connects a cell to the extracellular matrix. The attachment of a cell to the extracellular matrix or extracellular portion is regulated by these integrins. Now, what is the structure of this integrin? Integrin actually consists of two components, head and tail portion. The head portion, as you can see, is outside, okay, and the tail portion is present within the cell. The head portion, as you can see, it has certain divalent cation binding sites, and the head portion is actually that portion that attaches to the extracellular matrix, whereas the tail portion, it is actually binding to the cytoskeleton, actin, etc. And there are certain interacting protein like taline, filamin, alpha actinin. These are some of the protein that helps binding the integrin to the cytoskeletal elements. Okay, so integrin is also important, because if integrin is lost, then again the same situation will occur, that is the cell and the ECM attachment will be lost. Okay, so that is all that I had to discuss today. So to sum up, we have actually discussed the entire of the extracellular matrix, that is what are the different components, okay, the glycosaminoglycans, the different protein like collagen, elastin, fibronectin, okay, laminin, and then we finally discussed about integrin. Though integrin is not a component of extracellular matrix, but it is equally important because it helps connect the ECM component to the cell. So that's why I just in very short, I just mentioned about the integrin. So please go through this unit. Actually, it's a huge unit. It's given as a single unit, but I discussed it in two parts, in two sessions. The first part I focused on cell junction, and the second, today's discussion, I focused on extracellular matrix. Go through the unit. Again, if you have any doubts or you need clarification, you write to us or you contact us. We'll be there to help you. So go through the units and have a very happy learning. Thank you.